Hi everyone and welcome to today's Saturday session on Junior Cycle Science, where our topics for today are going to be photosynthesis and respiration. Now, these are two topics which are really important for Junior Cycle Science, and they do tend to come up as exam questions pretty much every single year. So today we're going to go through the theory behind both photosynthesis and respiration, and we're going to practice some exam questions as well, just so we know how we can pick up as many marks as possible in the final exam when we try to apply our knowledge to the questions. Now we're going to start off with photosynthesis, and for that we need to know the structure of flowering plants. So flowering plants are those that produce flowers, and we can see an example of that over here in the diagram. Now, we can split the flowering plant into two separate systems. We have our shoot system and we have our root system. So the part of the plant that grows above the ground is called the shoot, and the part of the plant that grows below the ground is called the root. And we can see that over here in the diagram. So we have our roots are currently growing underground and our shoot system is growing above the ground. Now the flower part is going to appear up here on the shoot, so we can see we have a couple of flowers, and we will also have some leaves forming on the stem or the shoot system as well. So for the structure of the flowering plants then, we can actually divide it up into four main organs. So very similar to how humans have organs, plants have organs too. Now, just on the levels of organization, we know the most basic level is the cell. Okay, they're the building blocks of everything. Moving on from a cell, if we have a group of cells working together to carry out a similar job, we're going to have a tissue. And then the organ, which is what we're focusing in on here, that's a group of tissues working together to carry out a similar job. Okay, so a group of tissues working together to carry out a similar job or function. So the four main organs we're going to look at, we have our roots, which we've already mentioned. That's what grows under the ground. Water can be absorbed by the root for the plant. We have our stem, which is part of the shoot system above the ground. We have our leaves, which we can see over here. They're involved in taking in and releasing gases. And then we have our flowers, which are located up here at the top. And they're usually bright and colourful. So if we have a look at the functions then of each of the organs, we're going to start off here with our root. Okay, so we have a particular type of root here, and its main function is to anchor and support the plant. Okay, so you can imagine the conditions that these plants are facing outside. It can be very windy, it can be very stormy, so they need to be able to secure themselves into the ground. Okay, so the plant or the roots are going to anchor and support the plant. Now, for photosynthesis, which we will come to in a couple of minutes, we do need to be able to absorb some water and also some minerals for the plant as well, and they're all going to come from the soil. So another function of the root is to absorb that water and minerals from the soil. The stem then, it's also going to support some flowers, it's going to support some buds, and it's going to support the leaves. And the stem is going to carry the water and minerals, which are after being absorbed from the soil by the roots, all the way up to the leaves and the flowers. Okay, so the stem is going to do that. In particular, it's going to be the xylem that does that, but we'll come to that in a couple of minutes. The leaf, that's what produces the food by photosynthesis, which we will come to. It's also responsible for gas exchange, okay, so releasing oxygen gas and taking in carbon dioxide gas from the environment, and also provides some protection for the plant as well. So one way it can do that is a lot of the time these leaves will have a waxy cuticle or a waxy surface. And what that means is if it was raining, the water will actually run straight off the leaf. Okay, so it's not going to weigh the plant down. It will just run straight off and make sure that the plant is going to be protected. That's just one way it can do it. And then we have our flower. So part of the flower, what's it going to do? It's going to produce pollen for sexual reproduction. And it's also going to attract insects to it so that pollination can happen, or they can take the pollen from the plant and deliver it to other plants. Okay, so that's the four main organs we have. We have our root, we have our stem, we have our leaf, and we have our flower. Now, we're going to focus in on the leaves for the next couple of minutes because this is the primary site of photosynthesis. Okay, so the leaves are the main site of photosynthesis in plants, with the chloroplast being the main structure involved. Now, this chloroplast contains this green dye called chlorophyll, and it's actually the chlorophyll that will absorb the sunlight for photosynthesis. Okay, so it's all happening inside the chloroplast, inside the leaf of a plant. Now, they have a number of adaptations to allow for it to take place efficiently. So we want photosynthesis to take place as efficiently as possible. So they're very thin. The leaves we can see down here, very thin. You even know that from yourself if you look outside, or if you go outside and take a leaf off a plant. If you hold it to the side, you can see that it's really thin. So chloroplast can absorb as much light as possible. Now, on the underside of a leaf as well, they have these tiny holes, which we refer to as stomata. And it's those stomata that allow gas exchange to occur. Okay, so they allow oxygen gas to leave the leaf and they allow carbon dioxide gas to come into the leaf. Now, if you want to see the stomata in the lab, one really easy way to do it is if you get a leaf and turn it over to the underside, so the non-waxy layer, get a bit of clear nail polish, coat the underside of the leaf in clear nail polish and let it dry. Peel it off after a couple of minutes, and when you view that underneath the microscope, you will see these small little holes called stomata. Okay, so they're responsible for allowing the gases in and out of the leaves. Now, photosynthesis itself, why are we focusing in on the leaf and what's so important about photosynthesis for the plants? Now, we are what we call heterotrophs. So as humans, we're heterotrophs because we get our food from other sources. Plants, on the other hand, though, are autotrophs. Okay, because they make their own food. So photosynthesis is the process by which green plants make their own food. So plants are autotrophs, they have to make their own food, and it's photosynthesis that they do that in. So the energy provided to the plant is in the form of glucose, so C6H12O6, that's the chemical formula for glucose, so that's the food that they're going to try and form. And humans also benefit from photosynthesis as well, because a byproduct of photosynthesis is oxygen gas, which is obviously what we use to breathe in. Plants are really important for the environment as well, because as part of photosynthesis, they actually absorb carbon dioxide into their leaves. So they're what we call a carbon sink, because they take carbon dioxide from the environment and in return they return oxygen to the environment. But the big thing here is they're trying to produce their own food, which is going to be glucose, and they do this by photosynthesis. Now, for photosynthesis to occur, there's 4 requirements. Okay, we have sunlight, we have chlorophyll, we have carbon dioxide, and we have water. The sunlight is going to come from the sun, and that's trapped by the chlorophyll inside the chloroplasts. So the chlorophyll is the green dye that's inside the chloroplast. We also need some water, so that's going to enter through the roots, and it's going to travel right from the roots up to the leaves through the xylem vessels. Okay, chlorophyll, that's the green dye that's in the chloroplast. It's what traps the sunlight. And then carbon dioxide, that's going to come from the air, it's going to come from the environment, and it's going to enter through the stomata. Now, just a quick point on the water entering the roots here as well. So how does the water actually move from the soil into the roots? It's through a process known as osmosis. And osmosis is the movement of water molecules from a region of high concentration to a region of low concentration. And it's through a semi-permeable membrane. So I'm just going to make a quick note on that. If you have something that's semi-permeable, it allows some substances to move in and out. So if you can imagine down here, we have some water molecules. We have our semi-permeable membrane, and we have water molecules on the other side as well. Now, because we have a high concentration of water molecules here on the left-hand side, they're going to try and balance out, and some of those water molecules are going to move over to the low concentrated region, and we should end up with a scenario where we have an equal amount on both sides. Okay, this is a passive process. It doesn't need any energy. It naturally happens where the water molecules will move from a region of high concentration to a region of low concentration. And that's exactly what's happening down here in the soil. Okay, if we were to water a plant, there's going to be a high concentration of water in the soil. It's going to sense in the roots that there's actually a really low concentration of water there. So through this process of osmosis, it will move through the semi-permeable membrane, which is the root, and it's going to move into the root where there was low water concentration. And then that water will move up through the stem to the leaves. The light is going to be trapped by the chlorophyll. We're going to have carbon dioxide coming into the leaf through the tiny holes underneath, which is known as the stomata, and the chlorophyll then is what's going to be absorbing the sunlight. So they're the 4 requirements, or the 4 things that we need for photosynthesis to happen. Now, what actually happens in photosynthesis? We're going to look at this in 2 ways. We're going to look at a word equation for photosynthesis, and then we're going to look at the balanced chemical equation. And it is important that we know both for the final exam. So for photosynthesis, the carbon dioxide, which is one of the requirements, is going to be taken into the leaf. We're also going to have water absorbed by the roots. The sunlight is going to be trapped by the chlorophyll, and we're going to produce glucose and oxygen gas as well. Okay, so we can see our 4 requirements here. We either sunlight, or chlorophyll, or water, and our carbon dioxide. So carbon dioxide and water will be used, the sunlight will be trapped by the chlorophyll, and we will form glucose and oxygen. Now, if we were to put that into a chemical equation, carbon dioxide is CO2, Water is H2O. We're going to keep sunlight and chlorophyll just as words. Glucose is C6H12O6 and oxygen is O2. Now, this needs to be a balanced chemical equation. And what we mean by balanced is we have to have the same number of carbons on both sides, the same number of hydrogens on both sides, and the same number of oxygens. And if we look at the minute, it's actually not balanced, because over here on the right hand side we have 6 carbons, over here on the left we only have 1. So if we wanted to balance that out, I'd have to introduce a 6 over here just to make sure we had 6 carbons here and 6 carbons here. Okay, if we look over here at the 12 hydrogens, over here we only have 2. So to get from 2 to 12, we'll have to have a 6 here as well. Okay, because 6 2s is 12. So now we have our 12 hydrogens here and our 12 hydrogens here. Now let's look at the oxygens. So currently as part of CO2, we have 6 O2s. So if we have 6 O2s, that means we have 12 oxygens. Here we have 6 H2Os, which means we have 6 oxygens. So 12 oxygens plus another 6 oxygens is 18. And over here on this side, we have 6 plus another 2, which is 8. Okay, now if I introduce a 6 in front of that O2, we suddenly go from having 8 to having 18, because here we have our 12 plus another 6 gives us 18. And if we were to double check, we now have the exact same number of carbons, hydrogens and oxygens on both sides of our equation. So that's our balanced chemical equation for photosynthesis, and it is important that we know both of those. So again, our 4 requirements: carbon dioxide and water, sunlight and chlorophyll, and plants are autotrophs, they're making their own food, so they're making their glucose, and oxygen is another product there as well, which is really important for us. Now, as part of photosynthesis, the transport of food and water is really important for the plant. So water obviously needs to be transported from the roots where it's absorbed right up to the leaves, which is where the food is going to be made as part of photosynthesis. But once that food is made in the leaves, it's not just the leaves that need the food. The whole part of the plant needs some food. So the food needs to be able to be transported around the plant as well. So water is going to be transported in these vessels called xylem vessels, and food, such as the glucose which is produced as part of photosynthesis, is going to be transported by phloem tubes. Okay, so we have an F sound in food and we have an F sound in phloem. Okay, so water is going to be transported in xylem, food is going to be transported in phloem. Now, with photosynthesis, obviously plants want to produce as much food as possible, and there are a couple of factors which can increase or decrease the rate at which photosynthesis is happening, and in turn the amount of food that's going to be produced. So one of these factors is the amount of light that's available, because the more light that's available, the more photosynthesis the plant is going to be able to undergo. If there's no light available, it's not going to be able to undergo photosynthesis, because light is one of the 4 requirements. The amount of CO2 gas is also going to be really important. So if there's a high amount of CO2 gas, it might be able to undergo more photosynthesis. But if there's an absence of CO2 gas, then it's not going to be able to undergo photosynthesis and make their own food, because carbon dioxide is one of the requirements. And again, the amount of water. So it's one of the requirements for photosynthesis. If there's more water, there's a chance that the plant can undergo more photosynthesis again. Okay, so as part of your study of junior cycle science, you actually would have done an experiment to investigate how one of those factors can impact the rate at which photosynthesis can happen. Now, most of the time, the factor which you would investigate is the amount of light that's present. And we're going to have a quick chat about how you can set up an experiment to do that and what you would have seen in the classroom environment, because it is actually quite a common exam question. Now, your setup for the experiment might have looked slightly different from this, but the idea would have been the exact same. So we're trying to investigate the effect of light intensity on the rate of photosynthesis. So we're going to increase and decrease the amount of light that this particular plant is getting. Now, the first thing we're going to notice here is we're actually using a water plant known as Elodea or pondweed. And the reason we use a water plant is because it can undergo photosynthesis underwater, and because it's producing oxygen gas as part of photosynthesis, it's going to produce these air bubbles. And we can actually count those air bubbles to investigate how much photosynthesis the plant is undergoing. The more bubbles, the more photosynthesis is happening. The less bubbles, the less photosynthesis. So what you would have done, you set up the apparatus like that. You would have put your light source a certain distance away from the plant. Okay, so let's say at the minute there we have that at about 1 meter away from the plant. We'd shine the light over on the plant and we'd let it acclimatize for maybe a minute or 2. Okay, it's important that you don't start measuring straight away once the light has gone on it, because the plant has to get used to the amount of light coming in. So you would count the number of bubbles produced then per minute. You would move the light a little bit closer, so maybe we would go as far as 80 centimeters. Again, let the plant acclimatize to the amount of light coming in, and then count the number of bubbles again. Move the light a little bit closer again, maybe to 60 centimeters, let it acclimatize, count the number of bubbles, and eventually you probably end up with your light source right on top of the plant. And once you've left it to acclimatize for a couple of minutes, you can count the number of bubbles. And what you should see is that the closer the light is to the plant, the more bubbles which are produced, which means the more photosynthesis is happening. And that's exactly what we can see in this diagram here. So the effect of light intensity on the rate of photosynthesis. So the distance from the plant is along the x-axis, the bubbles per minute is along the y-axis, and we can see that the closer the light is to the plant, the more bubbles are produced on average per minute. The further away we go, the less bubbles that are produced per minute. So that's telling us that as light intensity increases, so too does the rate of photosynthesis. You can kind of think of the opposite there as well. As the light intensity decreases, so too does the rate of photosynthesis. So you can do the exact same experiment by increasing or decreasing the amounts of carbon dioxide that's available to the plant, but the light one is much easier to do. It doesn't matter if you haven't done the light one and you did do the carbon dioxide one, or if you've only done the light one and you still have to do the carbon dioxide one. You've only have to do one of the experiments, and you can answer a final exam question on either the effect of light intensity or the effect of carbon dioxide on the rate of photosynthesis. So whichever one you've done in class is more than okay, and you would have ended up with pretty much the same results anyway, because the higher amount of carbon dioxide gas you give to the plant, the more photosynthesis would take place anyway. So we're going to have a look at a couple of exam questions then based on photosynthesis. And this first exam question here was just to complete the word equation. So we know that water plus carbon dioxide, there are two of the requirements. The sunlight is going to be trapped by the chlorophyll, and we're going to produce glucose and oxygen gas. Okay, so that was just writing down the word equation for photosynthesis. So this was part of a question. It was more of a question, a habitat study, but then they brought in the plants and photosynthesis as part of it as well. So students were given permission to remove some green plants from the habitat to take back to their school laboratory. They did this in order to investigate factors that affect photosynthesis. Imagine that you're one of the students. You've been asked to carry out an experiment to investigate how any one factor affects photosynthesis. Name one factor which could affect photosynthesis and which you might investigate. So the one we're going to look at here is light intensity, but there is scope there to do carbon dioxide intensity or the concentration of carbon dioxide as well, but I'm going to focus with light intensity. List two factors which you will keep constant or fixed during the experiment to ensure that it is a fair test. So we're going to make sure that the water is at the same temperature. And we're also going to make sure that the water volume is the same. So make sure the plant has the same amount of water. Okay, you could keep the carbon dioxide levels the exact same as well, because we're not going to change those. Write a suitable hypothesis for this experiment. So a hypothesis, I'll just make a quick note of this over at the side. That's an educated guess based on observations. Okay, so what do we think our hypothesis will be for this? Well, as the amount of light goes up, the amount, I should say the rate of photosynthesis will increase. Now we have to draw a label diagram of the setup of our experiment. So we're going to have our water basin or water trough. And inside that, we're going to have our LED. I'm going to put that inside a conical flask in here. And then above that, we're going to have a test tube or similar. Okay, this is going to be filled with water. Just extend that up slightly. And there's also going to be water inside the test tube here. Our LED or our pondweed is going to be down here. And that's going to produce bubbles. And the reason it's going to produce bubbles is over here we're going to have our ruler, which is going to record the length that the light source is away from the experiment. Okay, so we have our light source. It has to be a label diagram. We have a water trough in there as well. And inside that, we're going to have water. And the big thing that's being produced here is our oxygen bubbles. I'll just label those as well. Okay, so that's the setup. And obviously, if we're changing the light intensity, we're going to move the light source a little bit closer or further away. Okay, really important. If doesn't mention it there, but if you were to write out the steps involved in the experiment, it's really important to let the LED acclimatise to the amount of light that's being shone on it before you start to take any observations or any measurements, I should say, which is counting the number of bubbles per minute. Okay, so that was photosynthesis, and for the next couple of minutes, we're going to move on and we're going to look at respiration. So before we do that, we're just going to have a look at the diagram over here. We can see the process of photosynthesis is happening on the right-hand side. We have our carbon dioxide and our water. Sunlight is going to be absorbed by the chlorophyll and the chloroplast. The plant will undergo photosynthesis and will make its food, which is glucose, and also oxygen is going to be a byproduct of that as well. Now, oxygen and glucose are actually going to be used in respiration, and the carbon dioxide and water, which is used at the start of photosynthesis, is actually going to be the products of respiration. So these go hand in hand with each other. Now, we're not actually in the chloroplast anymore. We're moving over to the mitochondria. And instead of actually making food, we're releasing the energy from food. So respiration is the release of energy from food. And the glucose that was produced in photosynthesis is now the food that we use during respiration. Okay, the mitochondria is going to be the site of respiration. And there were two types of respiration. We have aerobic respiration, which is respiration which requires oxygen, and we have anaerobic respiration, which is respiration which does not require oxygen. Okay, so just to focus in on that, we're moving away from the chloroplast, which was where photosynthesis took place. We're now in the mitochondria, and we're looking at the food source, which was made in photosynthesis. We're now breaking that down to release the energy from food. Now, similar to photosynthesis, we need to know a word equation and a chemical equation for respiration as well. And we can see here that we require oxygen and glucose. And from that, we're going to produce carbon dioxide, some water, and the reason we undergo respiration is so we can release the energy from food as well. Okay, so glucose and oxygen goes to carbon dioxide, water, and energy. Now, the chemical equation, glucose is C6H12O6, oxygen is O2, carbon dioxide is CO2, water is H2O, and energy we just actually leave as a word, which is just energy. And we have to make sure it's balanced. It's going to be the exact same situation here as photosynthesis. It's not balanced at the minute because we don't have the same number of carbons on both sides. We don't have the same number of hydrogens on both sides, and we don't have the same number of oxygens on both sides. So exact same way as photosynthesis, if we just introduce sixes across the board, it will become a balanced chemical equation. Because here we have 6 carbons, we have 6 carbons over here, we have 12 hydrogens, we have a total of 12 hydrogens there because 6 H2s, that will give us 12. And we have 18 oxygens here, we have 12 of them there, plus another 6 gives us 18. So that's our balanced chemical equation. Okay, so the glucose, the oxygen, the carbon dioxide, and the water appear in both of them. The only thing that's additional in the photosynthesis one is we have the sunlight being trapped by the chlorophyll, whereas in respiration we have the production of energy because we want to break down the food to release the energy from it. Now, anaerobic respiration is the opposite to aerobic respiration in that it does not require oxygen for it to happen. Now, anaerobic respiration is going to produce some energy as well, but it's going to produce less energy than aerobic respiration. Now, when we do some exercise, we need more energy and respiration will naturally increase, but we can't always transfer enough oxygen quickly enough to all of our cells. And if this happens, lactic acid will start to build up. So you might have heard of lactic acid before. It builds up generally around your muscles, and it means that you're going to tire a little bit more quickly. So that's an example of anaerobic respiration, and it's sometimes referred to as fermentation. Now, it's not always going to happen just in muscle cells. It can also happen inside yeast. And if fermentation happens in yeast, we will get the production of ethanol or alcohol and carbon dioxide. So the alcohol business is heavily reliant on respiration because that's what actually produces the alcohol. It's a process known as fermentation. It doesn't require any oxygen. So if you were to compare and contrast aerobic and anaerobic respiration, one of the differences there is whether oxygen is needed or not. So oxygen is needed for aerobic respiration. It's not needed for anaerobic respiration. Aerobic respiration is going to produce lots of energy, whereas anaerobic respiration, there'll be less energy produced. And aerobic respiration is going to produce CO2 and water. So think of the chemical formula we seen a while ago. We had our glucose and our oxygen was going to be used, and we're going to produce CO2, water, and energy. Now, anaerobic respiration is going to produce lactic acid if it happens in muscle cells, but if we had respiration in yeast, we're going to produce alcohol and carbon dioxide. Okay, so there's our two types of respiration, aerobic and anaerobic. Okay, so similar to photosynthesis, you will also undertake an experiment to investigate factors that affect respiration, and there are a number of factors that can affect it. pH being one, temperature being another, and the type or quantity or concentration of nutrients being a third. Now, more often than not, it will be pH or temperature that you will investigate, and usually it's going to be the temperature experiment. So one way which you can investigate this is to set up apparatus like this. So you will have a water bath, you will set it to a particular temperature, and you will vary that from experiment to experiment. So you might start off at 30 degrees, then move to 40 degrees, and then move to 50 degrees. Inside our test tube here, we have a glucose solution with yeast. So glucose being the food source, yeast is going to feed off of that and try and release the energy from it. Okay, so what should happen in this case is we should get a layer of foam start to form up here in the test tube. And depending on how high that foam layer is, the more respiration is happening, the more energy that's being released from the food. Okay, so as we go from experiment to experiment, if we're investigating the effect of temperature, we have to change the temperature of the water. Everything else is going to stay the same. So we would have the same amount of glucose solution, the same amount of yeast, the same amount of water in the water bath. The only thing that can change is the temperature, and we would then measure the amount of foam that's produced in there in the test tube. And the results we would expect here is that as the temperature increases, so too does the rate of respiration. Okay, so we can see here we started off at 20 degrees, then went to 30 degrees, then went to 40 degrees. We're measuring the height of the foam that's produced. We had 4 centimeters of foam at 20 degrees, 6 centimeters of foam at 30 degrees, and we had 8 centimeters of foam at 40 degrees. Okay, so our conclusion there is that as the temperature increases, so too does the rate of respiration, so the more energy is released from food. So an earthworm takes in and uses up a certain gas during respiration. So name the gas the earthworm uses up during respiration. So for respiration, it's the food and oxygen. And then the gas that the earthworm is going to produce is carbon dioxide. Another exam question here. So mammals, including humans, use some of the energy they release from food to keep their bodies at a higher temperature than that of their surroundings. What is the normal temperature of the adult human body? Give your answer in degrees Celsius. So body temperature is around 37 degrees Celsius. And part 2 says, what is the name of the process by which mammals release energy from food? And a big focus of our session today was respiration, because that's the release of energy from food. Now, this is only part of an exam question, because we would have seen a very similar question a couple of minutes ago when we were looking at the photosynthesis ones, but this one is slightly different because we actually have to draw a chart to represent the data. So the results of Anne-Marie and Lisa's investigation are shown in the following table. So we have the distance of the lamp from Eladia and the average number of bubbles produced per minute. So we have 1 meter away, 75 centimeters, 50 centimeters or half a meter, 25 centimeters and 10 centimeters, and then this is the corresponding number of bubbles produced per minute. Now we have to draw a suitable chart to represent this data. A suitable chart for this will be a bar chart, and we're just going to write down some success criteria for drawing our bar chart over here to ensure that we get all of the available marks for this. So a couple of things that we need to keep in mind. We need to have a title, so an overall title at the top of the chart. We need to label our bars when they're drawn. We need to label our axes. We need to use a ruler and pencil. So that'll be difficult for me today, but we'll see how we can do that. And we need to have bars, need to be even widths. And we need to have even spaces between the bars. And really important as well, we have to have a consistent scale. Okay, so if we keep all of that in mind when we're drawing our bar chart now in the next couple of minutes, we should be good to go. So I obviously don't have a ruler, so this is going to be a little bit more difficult, but I'm going to have my distance from the lamp along the x-axis at the bottom and the average number of bubbles produced per minute along this side. So I'm going to draw my axes. And if I have a look at the numbers here, I have 12, 21, 27, 33. So I'm going to do that each box is going to be worth 3. So we have 3, 6, 9, 12, 15, 18, 21, 27, 30, and 33. Now, I'm actually only going to write every second number because otherwise it can be a little bit cramped and it just leaves it a little bit neater. But if you want, you can write in every number as you go. So you can put in 6, 12, 18, and so on. Now, the distance from the lamp, I'm going to put the shortest distance first because we're starting at 0 down here on the left-hand side and we work our way up to the biggest one. So we have 10, 25, 50, 75, and 1 meter then, or 100 centimeters. So our first bar is going to be for 10 centimeters away. And at 10 centimeters, we have 12 bubbles per minute. So I'm going to draw my first bar here. Again, you'd use a ruler here to make it as neat as possible. You can color it in if you want, you don't have to, but that's 10 centimeters away. Our next bar is 25 centimeters away and we had 21 bubbles. Again, you'd use a ruler. So that's our second bar. Our third one was 27. I'll do labels now in a minute. And then we had a 33 and a 33. So let's check our success criteria. Well, first of all, there I haven't labeled all the bars. So I've labeled all the bars, that's good to go. I haven't given it a title, so I need to go back and give it a title. So I'm going to say the effect of light intensity on rate of photosynthesis. So the title is there. Have I labeled the axes? No, I haven't. I need to go back and do that. So this is going to be the number of bubbles. This down here is going to be the distance. So that's done. Have I used a ruler and pencil? Well, I haven't, but I would have if I was doing it in my copybook. Are the bars even widths? They are, because they're all 2 boxes wide. Is there even spaces between the bars? There is, because there's just 1 box between each bar, so that's consistent. And do I have a consistent scale? I do, because each box here was represented by 3 along the side, okay? So I'm happy out with that. So that's the bar chart. It was just one suitable type of chart that you could use to represent that data. Keep in mind your success criteria when you are drawing those though, because sometimes these graph questions could be worth 12 or 15 marks, and we need to be as accurate as possible to ensure that we're going to get all of the marks available for it. Final question then, describe the relationship between the independent variable and the dependent variable in this investigation. So we can say here, I'm just going to write it up above the box, as light intensity increases, so too does the rate of photosynthesis. And if you're asked to say, how did you know this was the case? You can say it's because the number of bubbles increased the closer the lamp was to the experiment, or to the Eladia in this case. So that's everything we need on photosynthesis and respiration. And just before we finish up today, I just want to bring your attention to the ExamRevision website, where you can access loads more content, just like what we went through today. Now, when you go to examrevision.ie and log in, you can see that we have a wide range of subjects available. So for junior cycle, we have business, we have geography, we have history, we have home ec, Irish, maths, science, and lots more there for leave insert as well. Now for junior cycle science, which is what we were looking at today, and all other junior cycle subjects, you'll see that it's laid out this particular way. So we have video tutorials, we have quizzes, we have presentations, we have an exam builder, and we have a resource pack. So for the video tutorials, if we wanted to find what we were working on today, it's in biological world. We'll scroll down, it's in biological world 7, we can see there respiration and photosynthesis. And if you wanted to find out a little bit more about any one of those, all you have to do is click on the video, press play, and it will talk you through everything you need to know on that particular topic. Now, just underneath that, we also do have a quiz. So if we press on the MCQ quiz, we can see that we've 11 questions. We have to press an answer for each one, and at the end, we'll get a total score out of 11 in this case. So you'll find out straight away which ones you got correct, which ones you got incorrect, and you can do that quiz as many times as you want until you know everything there is to know about respiration and photosynthesis. We do also have some PowerPoint presentations there as well, so if you wanted to click into those, you could get some notes on the individual topics. And down here at the bottom, we also have our exam builder. So this has all of the past state exam questions, and it also has all of the past mock questions on these particular topics. So you can add whatever ones of those you want to your exam by pressing the plus button, and up here at the top, your exam will start to create. So you can download all the questions, download all the answers, and by the end of that, you'll have answered all of the questions around each of the individual topics, meaning you're fully set up for success in your final exam. Now, if you want to find out a little bit more about the website, just go to examrevision.ie. If you click on plans and pricing, you will see that you can sign up for a monthly subscription. The more subjects you sign up for, the cheaper it will be, and there's great discounts happening at the minute as well. There is also a free trial, so if you press the orange Start Free Trial Now button, you can test it out for up to a week, see if you enjoy it, and then can start to sign up for the monthly subscription. And we do also have a wide range of revision courses as well. So these take place during the midterm breaks, so during October, February, we have them at Easter as well, and then in the lead up to the exams in June. So keep an eye out on our social media for when these revision courses will be happening. The earlier you sign up, the cheaper it will be for you, and it will be a 4-hour, 1-day course covering all of the individual sections of the Junior Cycle Science course. So thanks a million for joining us today for today's Saturday session. Keep an eye out on our social media channels and our YouTube to see what other Saturday sessions are coming up in the future, and I hope to see you all again at a Science One sometime soon.